Biology. Hi, I'm Gail Fizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. Lesson overview for today. We're in Unit 6, Gene Expression and Regulation. The topics include 6.3, Transcription and RNA Processing, and 6.4, Translation. By the end of this lesson, you'll be able to describe the mechanisms by which genetic information flows from DNA to RNA to protein, describe how the phenotype of an organism is determined by its genotype, and predict the causes or effects of a change in or disruption to one or more components in a biological system based on biological concepts. So grab your pen and paper. All right, for our warm-up, which was last night's homework, and it was from the DNA structure and replication from Ms. Kate. And here we have researchers studying new viruses analyze the genetic material found in 4 different virus samples to determine the percent nitrogen-based composition of each virus. The data are shown in the table. So we have a table-based distribution in 4 nucleic acid samples. We have sample 1, 2, 3, 4, and then we have nitrogen-based adenine, cytosine, guanine, thymine, and uracil. And the question is, which of the following samples most likely contains a double-stranded RNA virus? Okay, so I'll give you just a moment if you're joining us for the first time. And if you did your homework last night, just sit tight and we'll go over the answer in just a moment. I'll get my clock going in the meantime. Okay, so we need to tear this table apart, and we can, if we're talking about RNA, then anything that has thymine is not going to be an option, right? Okay, and now we're down to 2. So let's look at the A's and T's and C's and G's and see if they are about the same. So here we have 21% adenine, and uracil we have 19%. Close, but maybe we could do better. And then cytosine and guanine, 33 and 27. So we're looking at our A's and U's and our C's and G's. So here we have adenine 27, uracil 27, cytosine 23, guanine 23. So that's my choice, is sample 1. And let's see if that's right. Yes, it is. I hope you chose A. All right, so before we get started with what we need to know, I need to give a shout out to Jack, who is in Miss Dio's class in Camp Hill, Pennsylvania, and he asked me to tell his teacher that the students in her class appreciate all the hard work that she is doing to help them. Thank you, Mrs. Deal. All right, so genetic information flows from a sequence of nucleotides in DNA to a sequence of bases in a messenger RNA molecule to a sequence of amino acids in a protein. So that's a general overview. So we're going to go from, whoops, still not what I want it to be. All right, so we're going to go from DNA, okay, and then we're going to transcribe to RNA, and then we're going to take that RNA and we're going to then end up with a polypeptide chain in a protein. Okay, so that's the sequence that we'll talk about them. All right, so first of all, RNA polymerases use a single template strand of DNA to direct the inclusion of bases in the newly formed RNA molecule. This process is known as transcription. So what's going to happen is a molecule, well, an enzyme, RNA polymerase, is going to unzip this DNA, and it's going to assemble a strand of messenger RNA, and it's going to add nucleotides to the 3' end. Okay, so it started at the 5' end, and it will add them at the 3 as it goes. All right, the DNA strand acting as the template strand is also referred to as the non-coding strand, the minus strand, or the antisense strand. Selection of which DNA strand serves as a template strand depends on the gene being transcribed. So let me share with you how I look at this. Okay, so the first thing that I can see right here, that's not what I want. All right, so the first thing I can see is that I have a 5' end, and there's my AUG. So if this is my messenger RNA, I know it's going to be almost identical to my coding strand, okay, of my DNA, for the exception of my U and T, okay? So this would be, there's an ATG and there's AUG. So this is my coding strand. Now my non-coding strand is then called my template strand, okay? Template strand. All right, so template. So my template then is also my minus strand, and this would be my plus. And then this is antisense, well, I can spell, antisense, and this is sense, okay? Now, depending on it, there's where I don't want you to, this sometimes happens in my room, this question comes up. Is it, so the top one is the coding strand and the bottom one? No, you can't look at it as top or bottom or left or right or whatever in the diagram. You have to look at the direction of the 5' and 3' ends of the molecule, and the gene can be on either side. It just depends on which gene is being transcribed at the time. All right, so the enzyme RNA polymerase synthesizes messenger RNA molecules in the 5' to 3' direction by reading the template DNA strand in the 3 to 5 direction. So, like I just said, it's going to read this is your template strand, right? And so this will be, your messenger RNA will be assembled to be complementary to that template strand. So here's T, A, U, etc. And it's assembled in 5' to 3'. So nucleotides are added at the 3' end. So anything that you're making new, okay? So 5' to 3' is your direction. Okay, everything starts with 5, ends with, and then is added on the back at the 3' end. Now that goes for messenger RNA. It also goes for when you're replicating DNA. All right, so this isn't the most ideal diagram, but I kind of took two from the test bank and kind of merged them together to say what I wanted to say with this. In eukaryotic cells, the messenger RNA transcript undergoes a series of enzyme-regulated modifications: addition of a poly A tail, addition of a GTP cap, excision of introns, and splicing and retention of exons. So we're going to start here with, so here is our DNA, okay? And if you'll notice, there are different parts to it. There are the white bars that I just covered up. Let me uncover those. Okay, those are the introns, and I tell my students the introns are going to stay inside of the nucleus, okay? And the exons are then going to be joined together to make the gene that's going to be, well, to make the messenger RNA that's going to go out and be read on the ribosome. So this has to happen. Introns have to be cut out, okay? But also we've got to put a 5' cap and a poly A tail on our, there's many, okay, 300 of those A's, and we're going to, because we have to be able to get out of the nucleus, and we have to make sure that no enzymes degrade us on the way to our ribosome. So now we've cut out the introns, they're inside, and we're only left with our 5' cap, our poly A tail, and our exons together, and some untranslated regions that won't be read into the, that won't make the polypeptide chain, won't be part of that. All right, so now after this is called messenger processing is what it's called, okay? So when you have messenger RNA processing, I've got to get off of these pinks, they're not showing up good. All right, messenger RNA processing, okay? So it's not true mature messenger RNA until these steps have happened. And so now that's the processing, it happens inside of the nucleus. All right, so, and speaking of, this isn't the ideally what this image doesn't ideally match what I'm saying, but I think that you'll get it. Excision of introns and splicing and retention of exons can generate different versions of the resulting messenger RNA molecule. This is known as alternative splicing. I'm going to work backwards with this. So here we have this receptor, okay? Here we have this receptor, and it the ligand that will fit in it will be here it is right here, and that will go right there. Now, it was coded for by this strand of messenger RNA, which is different than this strand of our messenger RNA, because this messenger RNA will code for this receptor that will bind to this ligand. So now these are totally different, and they came from the same gene, but what's being cut out, see, we have segments 1, 2, 4, and 5 in this, so we used that one, those. And so those were the exons that were put together to make this messenger RNA. And 1, 3, 4, and 5 were the exons that were put together for this. So 2 was not an exon, it actually became an intron to do this version, is what I'm trying to say. All right, the sequence of the RNA bases together with the structure of the RNA molecule determines RNA function. Messenger RNA molecules carry information from DNA to the ribosome. Distinct transfer RNA molecules bind specific amino acids and have anticodon sequences that base pair with the messenger RNA. Transfer RNA is recruited to the ribosome during translation to generate the primary peptide sequence based on the messenger RNA sequence. Ribosomal RNA molecules are functional building blocks of ribosomes. So these are just the players. Okay, I'm going to go through the whole process on different slides, but these are the players involved. We're going to have our messenger RNA. Here's our 5' end and our 3' end. We're going to have our ribosome, which is made up of ribosomal RNA. Okay. So ribosomal RNA that's going to interact with our messenger RNA. This is our transfer RNA. It's going to bring in our amino acid. It has anticodons that will be complementary to eventually codons on this messenger RNA on the ribosome. Okay. All right, so we're going to start from the beginning. The salient features of translation include: translation is initiated when ribosomal RNA in the ribosome interacts with the messenger RNA at the start codon. And the sequence of nucleotides on the messenger RNA is read in triplets called codons. So here we have our small subunit, and we have our messenger RNA has engaged with this ribosome, the small subunit, and now the transfer RNA. So this was AUG, this is our start, and this is UAC, which is the complementary, A to U, U to A, G to C, and this is going to bring in methionine. And now that all these players are together, then the large ribosomal unit will join them, and now we have our polypeptide factory in place. Okay. All right, each codon encodes a specific amino acid, which can be deduced by using a genetic code chart. Many amino acids are encoded by more than one codon, and we'll talk about that a little bit later. All right, so let's start from the beginning. So here's our messenger RNA. And we can break this down into codons now, okay? So there's AUG, CCG, CAA, UCU, GUU, CAC, GCA, CUC, AUG, and then I don't have enough to complete there, so we'll stop there. Now, so this will be our AUG, and this will be our CCG, and then we have CAA, and UCU, and GUU, and where are we? CAC, and GCA, and I'll stop in a minute. GCA, CUC, and then AUG. Okay, so so we had AUG. So AUG, we went to our genetic code chart, and we can see that here's AUG. So this amino acid that was brought in, okay, was in fact methionine, and it would be this one right here, okay? I'm giving for the orientation of where the amino acids are on this strand. Okay, so then that was the first one. And then the second one, the CCG, we'll find it, CCG, then when we have proline, and that would be proline would be right here. Okay, so that one's done. Now CAA is glutamic acid, and so we can add this. Okay, that's done. And now presently in the P spot of the ribosome, okay, we have UCU, and we can make it that color. And now the next one that's coming in, now we can do a little bit more work with this. It's GUU. So now we have GUU, and there's valine, okay? And so this is valine. And now this anticodon that was complementary of this would be CAA, right? Okay, so what I don't want you to do is try to find the amino acid with the anticodon, okay? You look for it with the codon. Okay, that's enough on that one. I think that hopefully you got it with that if you are having trouble with the genetic code. All right, translation involves energy and many sequential steps, including initiation, elongation, and termination. Transfer RNA brings the correct amino acid to the correct place specified by the codon on the messenger RNA. The amino acid is transferred to the growing polypeptide chain. The process continues along the messenger RNA until a stop codon is reached. The process terminates by the release of a newly synthesized polypeptide and soon to be protein. Okay, so here we have our ribosome, our large unit, small unit, everything's that we needed to get started. Here's our first transfer RNA. It's going to take place. If you remember, we had spots. We had an arrival, a polypeptide spot, and an exiting spot. This comes in, 5' in, heads in this way. That's why it's over here on this side. Okay, so now we have this amino acid. The next one will come in. Let's go to step 2. So now we have 1, 2, 3, 4. We're on our 4th one. Now this empty transfer RNA will move out of the exit site. This one in the P site, the polypeptide chain will then covalently bond to the amine end of this amino acid, and you'll see that then the blue one will be, this will move here, and then that will all be strung together like this right here. So as we come in, this attaches, this bonds with this moves over, and then the other one moves out. And now we have our polypeptide chain, and now we have a release factor that has been coded for with one of those 3 stop codons. There's no amino acid coming in with the stop. So this will be released and then go off for further processing. All right, translation of the messenger RNA to generate a polypeptide occurs on ribosomes that are present in the cytoplasm of both prokaryotic and eukaryotic cells and on the rough endoplasmic reticulum of eukaryotic. Okay, so here we have, here's some ribosomes, and these are in the cytoplasm. This is a eukaryotic cell, and then we have some on the endoplasmic reticulum also. And then, so that's eukaryotic here. We had them in 2 places, endoplasmic reticulum and in the cytosol. And now here we have them in our prokaryotic cell, okay? And I'll talk about this in just a second. All right, in prokaryotic organisms, translation of the messenger RNA molecules occurs while it's being transcribed. So there's no nucleus, there's not processing, but we do have transcription going on, and then immediately we can go into translation for that protein to be synthesized. And it can happen on, and it happens on more than one ribosome at a time. All right, nearly all living organisms use the same genetic code, which is the evidence for the common ancestry of all living organisms. Whether, I always tell my students, whether you are, you know, our cat or our dog or a fish in our house, that we could be our genetic code. If we had a CUC, it's still going to be leucine. All right, genetic information in retroviruses is a special case and has an alternate flow of information from RNA to DNA made possible by reverse transcriptase, an enzyme that copies the viral RNA genome into DNA. The DNA integrates into the host genome and becomes transcribed and translated for the assembly of new viral progeny. So here we have our virus, and we see that the envelope plasma membrane is going to become part, the plasma membrane will be fused with the plasma membrane of this cell, and then it's going to empty its contents, and that you could see that the virus has these protein capsids. The little black line is the RNA. The little green is going to represent our reverse transcriptase enzyme. And now let's look at that. We have, here's our RNA, reverse transcriptase, and with that reverse transcriptase enzyme, we can make a strand of DNA. And then we'll have a strand of DNA have a complementary strand of DNA. And now once we have a double-stranded DNA, then we can be inserted into the genome, and then messenger RNA can be transcribed, because the point is to make more viruses. So now we need more membrane parts, we need more protein receptors, we need more capsids, we need more enzymes, we need more of everything before we can exit the cell and leave. But the point of the retrovirus part is that it started out as RNA, and then it made a strand of DNA, and then it made a strand of RNA again to make the protein, right? Now, usually we start with DNA and we go to RNA, and then we have protein. So we did things a little bit backwards than usual, and that's where the retrovirus comes from. All right, so check for understanding. Here's a question. Figure 1 represents part of the process that occurs in eukaryotic cells. There are untranslated regions, UTR, in this sequence. So here's what we're looking at. We have a 5' cap, we have poly A tail, we have some exons, we have all the parts we need, and we can see that we can see that introns have been cut out of this strand of DNA, okay? All right, so which of the following best explains the process represented by figure 1? A, the synthesis of messenger RNA in the 5' to 3' direction. B, the modification of a protein to produce a functional form of that protein. C, the translation of messenger RNA molecule into a polypeptide. Or D, the enzyme regulated processing of pre-messenger RNA into mature RNA. So it had nothing to do, we're not synthesizing a new strand right now. We're already past that. And we're not at the modification part that's after translation. And then we're not translating, we're not on ribosome right now. So now this is our answer. That's what's going on. This was pre, and now we have mature, and we're ready to go to the ribosome. Okay. All right, so a lot of the questions today are going to have to do with changes. So this was the most appropriate science skill to talk about. Predict the causes or effects of a change in or disruption to one or more components in a biological system based on biological concepts. We're going to do some calculations, very simple. Provide is a word that might come up. Predict, we're going to describe. Remember, it's describing, just list the characteristics, and there's one. There's a certain describe that I want us to take us back to what describe really means. And then explain is the how or why, and justify, claim, evidence, reasoning, okay? All right, so Loeys-Dietz syndrome, type 2B, is a human genetic disorder associated with mutations in the LDS2B gene. Researchers have proposed that the LDS2B gene produces a growth factor receptor protein that regulates cell signaling pathways. The predicted product of the gene is a 565 amino acid protein with structural regions that bind to signaling molecules, span the plasma membrane, and have protein kinase activity. Protein kinases are cellular enzymes that transfer phosphate groups from ATP to target proteins. A representation of the LDS2B gene is shown in figure 1. So we can see that we have a start to our gene and a stop, and we have a transmembrane region and some kinase regions in this dark shaded, and then in the lighter is signaling molecules. Okay, figure 1, genomic structure of the LDS2B gene. The 7 exons, so that's what we're talking about right here. These boxes are the exons, and so those little lines would then be the introns. The 7 boxes, numbered rectangular boxes, the introns, then lines between the boxes, and the start and stop signals for translation are represented. The portions of the gene that encode the different structural regions of the protein are shown, and the location of a of a methionine to valine substitution at position 425 of the encoded polypeptide M425V is indicated. So we're going to go, there's going to be a change, okay? We're going from methionine, remember this because when I turn the page, methionine to valine. Okay. Now we have our genetic code. Let's calculate, first thing we have to do, calculate the minimum number of nucleotides required in the coding region of the LDS2B. So minimum number of nucleotides, okay, to produce and terminate the 565 amino acid polypeptide. And I'm going to stop right there. So let's take, I'm going to just think out loud, 565, and that is how many amino acids we have, right? So how many codons did it take to do that? It took 3 each. There's 15, 18, 19, 16, am I right? 1695. Okay, now it set up a hint word here, and terminate. Even though we don't have an amino acid, right? We don't have, there is still something that's needed to code to stop. So let's add another 3 to that. Okay, so let's keep that number 1698. This is simple math. You know, that there still could be simple math on your exam. You don't need a calculator and you won't be graphing, but simple math could actually still be there. Provide 2 reasons that the number of nucleotides in the mature messenger RNA may differ from the number of base pairs in the genes. Okay, so we're talking, I'll give you just a second to think about that before I start adding in. Okay, so what did you think about? Did you think about when we cut out the introns? Right, we cut them out. So there were introns in the DNA. So that would be a difference. We added a 5' cap. That would be a difference. And we also added a poly A tail. All right, that would be a difference. All right, so that's all I can think of, right? I think there's probably 6 or so options. I just can't remember them all. All right, one genetic change associated with the disorder results in methionine to valine substitution at amino acid 425, as shown in figure 1. Using the codon table in figure 2, predict a DNA point mutation that will result in methionine to valine. So let's look at, what did it take to make a methionine? It took this, right? AUG. And we ended up with valine. So valine could be any one of these, but what's the closest to it that only one single point could be changed? It could be this one, right? So if it was GUG, then it would be valine. So this one letter. So we could have just a change from A to G, or if this was the DNA, and because this is the messenger RNA, remember? So this would have been a T to a C, okay? All right, so let's finish up. I think we might be finished with this question. Let's see. Here we go. So here's the first part. Did we say 1698? Yes, we did. Response demonstrates understanding of the triplet code of the polypeptide, and response demonstrates understanding of the role of the stop codon in terminating translation. So there's our 1698, and there's 1698. All right, the next part, we did say that it was processed and introns were removed, and that a 5' cap was added, and poly A tail adds nucleotides to the 3' end that are not encoded by the gene. We didn't talk about this, and we didn't talk about that. But what I wanted to make a point to say to you is this work that I do with you here, this is my planning work. So if I plan before I start writing, and then I put it into full sentences that are complete thoughts, okay, that bring all the pieces together. Like when we said that it has a 5' cap. Here is 5' cap adds a nucleotide that is not encoded by the gene. You should say more than just 5' cap, okay, because that would be more like an identify answer. Now let's look at the bottom. We have A to G because it was AUG to GUG, and also you could have said T to C, or you could have said the combination of the two, the A/T or the G/C. All right, now for a short FRQ. I know this one looks a little bit different than usual, and I got it right out of the CED. There's two FRQs in the back. There's a short one and there's a long one, and I put a copy of the short one is in I think maybe 2 lessons ago in one of the Power slide decks that I didn't have time to get to in the presentation. But anyway, they're both in the CED, so they're not a secret. They're available to everybody. All right, so in many countries, Anopheles gambiae mosquitoes are responsible for transmitting the parasite that causes malaria to people through their bites. A primary tool for mosquito control is the use of insecticidal nets sprayed with chemicals known as pyrethroids, which are relatively safe for people but toxic to mosquitoes. However, mosquito resistance to pyrethroids have now become widespread. Pyrethroids interfere with the function of a transmembrane sodium channel found in the cells of mosquitoes, and that's in figure 1. In one common mutation to the channel protein, a phenylalanine is substituted for a leucine at amino acid position 1014. Scientists hypothesize that this mutation is responsible for some cases of pyrethroid resistance. Figure 1 is a schematic drawing of the transmembrane sodium channel targeted by pyrethroids and other insecticides. The arrow points to the position of the amino acid 1014. To investigate pyrethroid resistance, mosquitoes were collected 4 times over a 2-year period from the following regions. In region A, a southern vegetable growing region where large amounts of insecticides are, okay? And region B, a northern rice growing region where very little insecticide is applied. Okay, let's see what else. Scientists exposed the collected mosquitoes to filter papers soaked in 2 different pyrethroid insecticides, deltamethrin and permethrin, and the percent mortality of the mosquitoes was determined after 24 hours. The scientists simultaneously determined whether leucine or phenylalanine was encoded at position 1014 by each of the 2 copies of the sodium channel gene. All right, so now we have a table. It looks a little bit different because now we have our bars heading this way. Now, look, here's percent mortality, and here is our regions. We had a region that had a lot of insecticide use and then one that didn't, remember? And then we have our months. So figure 2, susceptibility of A gambiae mosquitoes from 2 regions to the pyrethroids, deltamethrin and permethrin. A mosquito strain that is acceptable to the insecticides displayed at least 95% mortality in all experiments. Mosquitoes exposed to untreated filter paper displayed less than 10%. So it's giving you some additional details besides what's there. Error bars represent standard deviation, and ND means no data are available. All right, here's another table. Frequencies of leucine and phenylalanine at position of 1014 of the sodium channel is looking to see if, here is in region A, and it looks like most of them are homozygous for phenylalanine. And then B, it looks like there is a combination, but most are homozygous for leucine. Okay, here's our job. Describe. Okay, we're going to describe the most likely cause of the amino acid substitution in the sodium channel protein. Okay, so A. Think about what you think A is. Don't overthink it. Okay, in my planning, I'm probably going to think that there is now this is describe, most likely cause. So there could be a mutation or a change in what? What am I talking about in my description? In the DNA, right? Okay. Explain how the substitution of a single amino acid in the channel protein could cause pyrethroid resistance in mosquitoes. Okay, so now if we had this substitution, what could happen? So if we have this substitution, we could have a change, right? We could have a change in our amino acid sequence, which could mean a change in our protein, which means a change in the way it folds, which means a change in the way something can bind or can't bind, which changes its function, okay? Identify the dependent variable in the experiment whose data are graphed in the figure 2. So let's go back to figure 2 and find the dependent variable. So let's get rid of some of all this. So what do you think is the dependent variable here? Should be here, but that's what's we're changing. So this is kind of flipped, right? So here is our dependent variable, and there is our, and this would be what we're changing. The two, it's kind of flipped. All right, so if we just stood it on its side, it would be fine. All right, now what's the other? Whoops. Now identify the positive control in the experiment, and let's find that. It told us that. It said a mosquito strain that is susceptible to insecticides displayed at least 95% mortality. So that's what it looks like when it is susceptible, okay? And then the next thing, justify exposing some mosquitoes to untreated filter paper each time the experiment was performed. Remember, there's some things you should write about when we're talking about what? The control. It's the control, that's number one, and then there's no treatment, and then that whatever is happening in your experiment is happening because of something else and not because of the it's happening because of the treatment, not because of the filter paper in this case. There's an L. Okay, now let's check and see how we did. All right, A change occurred in the DNA sequence encoding the sodium channel polypeptide, okay? Now you should have taken that sentence that we talked about, and we said mutation, change in the DNA. Okay, did a pretty good plan on that one, but I didn't go as far as saying what I was changing, what ended up changing. Okay, explain how the substitution of a single amino acid in a channel protein could cause pyrethroid resistance by mosquitoes. The substitution could change the shape of the protein channel so that pyrethroids can no longer affect it, bind to it, or interfere with its function. Identify, we got that. Identify the dependent variable, percent mortality. Okay, identify the positive control, testing the strain that is acceptable to the insecticides. Justification of exposing some mosquitoes to untreated filter paper each time the experiment was performed. Okay, it says exposing mosquitoes to untreated filter paper confirms that any observed mortality is from the insecticides rather than the filter paper, okay, or any other experimental conditions. Okay, there were two other parts to that question in the back of the CED, but they had to do with evolution, and so they went into topics that were not included in the units 1 through 6, but you could go find them. All right, so here is one more. A processed eukaryotic messenger RNA lacking introns is introduced into bacteria so that the bacteria can produce a large quantity of the encoded polypeptide. Translation of the messenger RNA to produce a polypeptide involves the binding of large and small ribosomal subunits to messenger RNA. Each complete ribosome has 3 tRNA binding sites that align with 3 sequential codons in the messenger RNA. 3 steps in the process of translation elongation, the addition of amino acids to a growing polypeptide chain are illustrated in figure 1. So if this is figure 1, you've seen it, I've used it actually a number of times already through these videos. A schematic drawing of 3 steps in translation elongation. Sequential codons are numbered, and AA, okay, right here, represents an amino acid. So the first thing we need to do is we need to describe why translation of a messenger RNA can start before transcription of the messenger RNA is complete in prokaryotes, but not in eukaryotes. So I'll give you just a moment to think about that. Okay, and should go ahead and jot some thoughts down. And then referring to figure 1, explain the process illustrated between step 1 and step 2. What happened in step 1? We had this polypeptide chain is going to be then covalently bonded to this next amino acid that's coming in. So there we have 1, 2, 3, 4. 1, 2, 3, and there's 4. And so when you explain that, you need to say amino acid 3, it was then joined to amino acid 4 on the amine end covalently bond, and then this is going to move to the exit site, and this is now going to move to the polypeptide site, and then the next amino acid. Okay, do all those details when you explain what's going on here. All right, let's check. I think that I have C and D before we get to the scoring guidelines. Okay, this is my favorite part. All right, fill in the boxes in the template to identify the nucleotide sequence from 3 to 5 of the transfer RNA anticodon that will recognize codon 5 and the transfer RNA anticodon that will recognize codon 510 of the messenger RNA template, and to identify the amino acids that will be added at these 2 positions. So I know we've already did this like 10 times, but we're going to do it 2 more. So here we have a CGG, that's our amino acid at location 5. We're going to go C, and then G, and then G. And what is that? It's arginine, right? Okay. And so that took, what had to bring that in? It had to be a GCC. Now at position 510, there is going to be a UAG. So now let's look at U, A, and G. And now we have a stop. So UAG is then going to have nothing, right? There's not going to be an amino acid. There's not one. And that anticodon then would have been what? It would have been the UAG, so it would have been A, and I can't think for some reason. There we go. All right. Did I do that right? Okay. So there we have stop. That's the point. Stop's coming in there. All right, now let's check. The response indicates that the prokaryotes do not have a nuclear envelope that separates the genome from ribosomes in the cytoplasm of the cell, whereas where eukaryotes do. I hope that you said that. The nuclear membrane was an essential part of that sentence, separating the genome from ribosomes in the cytoplasm, and in eukaryotes, but the prokaryotes do not have to do that. The response, and then B, the response indicates that the growing polypeptide chain attached to the transfer RNA aligned with codon 3 of the messenger RNA is transferred to the amino. See, that's what I said you needed to say. All of those features attached to the transfer RNA aligned with codon 4 of the messenger RNA. And then 5, we said was arginine, right? And then 510, that's why I knew that. I wanted my little, what do you call it? The indicator to stop. There's not an anticodon bringing in a stop. There's no stop, okay? All right, and then D, the response indicates that the polypeptide produced by translation of a processed eukaryotic messenger RNA by a eukaryotic cell or a prokaryotic cell into which it has been introduced should have the same amino acid sequence because the genetic code is almost universal and the same codon chart can be used for both prokaryotic and eukaryotic organisms. Okay, I hope you have time to go back and look at this question. I really like this question. I think it's a good learning tool to learn how to do protein synthesis and how to read the codon chart, one of my personal favorites. All right, debrief and summary. The takeaway for the week: heritable information provides for continuity of life. I know that you're able to do all of the learning objectives for the week, and I know that we are constantly on this predict the causes or effects of a change, but that's super important. It's one of your more difficult science practices. Common errors and misunderstandings. Don't refer to DNA as the bottom or the top. Each has a role depending on the gene that is being transcribed. And then use the codon on the genetic code chart, not the anti-codon. And now your homework for tonight, you have one question, and you're going to look at figure 1, model of process involved in gene expression. Which of the following best explains what strand X represents? And you have 4 options. A complementary RNA sequence because it contains thymine. B, the coding strand in this process because it's being read 3' to 5'. C, the antisense strand because it is serving as a template. And D, the pre-messenger RNA because it does not yet have a GTP cap. So you do that for your homework, and Ms. Kate will go over that first tomorrow. All right, so before I let you go, and look at this, I have a few things I want to say after this slide, so don't go. All right, so your to-do list, check your device and internet, okay? Check your email address, phone number. I have more to say about those, or you have more to read about those. Tell your teacher you're testing. Practice questions, okay? That green button. And be sure you do your homework. And then Ms. Kate will be with you tomorrow evening for gene regulation. And now I want to go over just a few slides. I put this together this morning after this was released yesterday. So if you go to cb.org, you'll get to this page, okay? And then you click on this blue up-to-date information, and then that's going to take you to this page. And you could go either way, depending on if you're a student, you're not banned from this updates for educators, okay? This information, if you're not able to contact your teacher, you need to be reading it yourself anyway. So updates for AP educators, and then learn more about taking the exam, and then click on this 2020 AP testing guide, and it is this fantastic, very clear, concisely written testing guide, okay? There's not even though it says that looks terrible, right? But it's not. It has simple information and diagrams on each page to walk you through. I walked through my students in 2 periods this morning I put that on the presentation like you see me present on, and then we walked through each slide and let them ask questions. The most valuable thing that I can tell you in here is a May 4th practice, like a mock submitting a question. That's the day that is, to me, going to be the most important. But go find that file, read through it, and then that can answer all of your questions. And I think that's it for today. All right, thank you.